Okay, so first speaker, our Justin, I think can take it away.
All right, thank you. And yeah, thank you for having me here. So I'm excited to talk about a lot of work that I did during my PhD that I just received last year at ASU. This was something that I spent a lot of time trying to rack my head on, figuring out what are some good ways to actually characterize high contrast image scattered light debris disk systems. And you'll see why this was such a topic of frustration for me for the past couple of years throughout this talk. Okay. No, is that not going to work? Okay, so for those of you not familiar with debris disks, we can think of these as sort of a midway stage of the sort of planetary formation process. So some of you who may work on protoplanetary disks, you may be familiar with these massive flared structures full of gas and dust. These are very, young. And as time goes on, some of that gas starts to evaporate and dissipate, and the dust grains in your disk start to settle down more into the midplane of your system. And this keeps going on until you've pretty much lost all of your gas, either due to photoevaporation or accretion. And what you're left with are the dust grains. And that's this debris disk stage that I'm going to be talking about. Okay. To put it into some more context, our own solar system actually has a few different dusty components to it. So you may be familiar with the asteroid belt. You can think of this as like a warm inner belt. And we also have the Kuiper belt, which is a sort of outer cold belt. A lot of the systems that I'll be talking about today are mostly these outer belts. So very cold, cold dust. If you're interested in learning more about debris disks, I highly recommend this recent review paper that came out by Tim Pierce. It's a very good introduction to to what these systems are and how we observe and how we model these systems. So definitely check that out if you're interested. Now, observationally, the first debris disks was actually detected in the 80s with the IRAS satellite. And the way we first found these were from noticing these excesses of infrared emission around stars. So this is from Allman et al. 1984, and this is showing this really intense infrared excess around Vega, which, Vega is a really bright high mass star. You don't expect this much infrared emission compared to the stellar photospheric emission. Since then, we've been able to observe debris disks in a lot of ways, including imaging. So this is a figure from Luca Matra's paper. This is an ALMA observation of the debris disk around Vega, and you can see that you can resolve this sort of belt or ring type structure. Okay. But for a long time, these sort of indirect methods of finding infrared excesses have sort of been the primary way which we've observed and detected debris disks. So here's another figure from Tim Pierce's really nice review showing the spectral energy distribution. This one is actually of the Beta Pictoris system. And, we have all these measurements from various observatories such as Spitzer and Herschel that just show, all of this excess infrared emission at these mid to far infrared wavelengths. And you can actually, from these indirect measurements, you can actually start to characterize some of these systems. So you can get a sense of the temperature, which gives you a rough idea of the location of the dust belt in a system. So that's what this figure is trying to illustrate here. So you have these this really big outer disk component, and then you have some contribution from an inner disk component as well. Now, these indirect measurements are really good, and it's really good for building a sense of the general population of these types of systems. But what I'm really interested in is the actual structure, so the bell-like structure of these systems. And to get at that, you really get the most clear picture from doing resolved imaging. And so this is an infographic, or this is comparing observations of the debris disk around Fomalhaut and at all these different wavelengths. So we have the visible with HST STIS, all the way to the millimeter with ALMA continuum imaging. And yeah, you see this like really nicely resolved ring type structure. Now, for this talk, I'm mostly going to be focused on this sort of visible near-infrared regime, specifically the near-infrared, but this all sort of probes this scattered light regime of these debris disks. So these dust grains in the debris disks in the sort of mid-infrared to longer, the dust grains are absorbing photons or absorbing energy from their host star and re-emitting that energy as thermal emission. But in the visible and near IR regime, it's more efficient to actually scatter the photons off of the dust grains rather than absorb and re-emit in thermal emission. Okay, and so part of why I'm interested in imaging in general is that, being able to see these resolved structures can allow you to learn a lot of interesting things about these systems. So, not just in terms of ring sizes, but you can start to probe things like the vertical structure in more edge-on systems. And you can also start to look for sort of these asymmetric features or departures away from circularly symmetric. And these are interesting because, these sort of asymmetric features could be induced by dynamical interactions such as perturbations from planetary companions. So especially these sort of like carved narrow ring type structures, you can imagine some sort of like Jupiter mass companion might be carving out an inner edge to some of these debris disks, or you might have a planet on an eccentric orbit that might be inducing like a warp type structure or a misalignment between different dust components in a debris disk. Now, part of what interests me about the scattered light regime in general is that you can start to probe this property known as the scattering phase function, which I'm going to get to in some later slides. And something else that's really interesting is being able to probe polarization as well. So I'll talk about it in a couple slides, but we have these really nice high contrast imaging instruments that are also capable of doing polarimetric observations. And the dust grains in the disks, in addition to just scattering the photons, they also linearly polarize them as well. Okay, so here's my sort of high contrast imaging quick overview. So again, we're focused in the sort of near infrared regime, but at these shorter wavelengths, stars are really bright, and it's really hard to resolve these sort of nice structures in these disks that can be anywhere, 10,000 times or 100,000 times fainter than the stars that they actually orbit. So some important words here, contrast. This is just explaining sort of the brightness ratio between like any astrophysical object, like a planet or a disk, compared to a host star that it surrounds. And I'm not going to go into too much detail about these other terms because they're not super relevant, but these are also important for doing high contrast imaging. So having adaptive optics to help really focus your starlight, as well as a coronagraph, which blocks out a lot of the really bright starlight so that you can actually see things that are, 10 to 100,000 times fainter than the star itself. Okay, and so a lot of the observations and analysis that I've done come from observations from this imager, or this instrument called the Gemini Planet Imager. This was a near-infrared high contrast imaging instrument that was installed at Gemini South up until 2020. And some of the key characteristics is that it has a spatial resolution of about 14 milliarc seconds per pixel. And we can probe down to about 100 milliarc seconds in projected separation. It uses one of these coronagraphs, like I mentioned, And peak contrast can reach down to about 1,000,000 times fainter than the target star that we're looking at. And it's also capable of doing integral field spectroscopy and polarimetry. So that means that you can get spatially resolved spectra and spatially resolved polarimetric images from whatever systems you might be looking at. So, as you can tell from the name, the primary goal of the Gemini Planet Imager and other high contrast imaging instruments such as SPHERE were to search for these directly imaged planets. But if you're familiar with high contrast imaging, maybe you know that both of these instruments have definitely resolved and detected a lot more disks than planets, which is good for me because I really like disks. But this is just sort of a sample of quite a few disks that GPI has been able to image. And this is a figure from one of my frequent collaborators, Thomas Esposito. And yeah, these are all in sort of the debris disk stage for the most part. I don't think there are any younger disks in here. Yeah, no, these are all the debris disks. But yeah, we have this really nice sample of spatially resolved debris disks for which we can start to probe some of these sort of dynamical and morphological properties of these systems. So what I want to talk about in my talk is, actually starting to get at some of these bigger studies of multiple systems all at once. So kind of doing a quasi-population analysis. And some of the problems that go into this is that there have been several papers across the literature that talk about some of these high contrast imaged resolved debris disks, but they often have to perform their own sort of like tailor-made approaches and analyses, and it's really hard to do sort of the apples to apples comparison between all of these different papers, to do this big population analysis that I want to do. Another big motivating factor for my study is that when it comes to characterizing some of these debris disk systems, there are a lot of limitations and a lot of really bad limitations and assumptions that have made characterizing these systems really difficult. And all of this is to sort of get at my goal of doing this uniform analysis of this scattered light debris disk population. So I'm going to go over a few of these points first. So yeah, I already mentioned it's difficult to compare all of these single studies of these different debris disks, as you're not comparing apples to apples, really. And this is most critical when it comes to understanding the modeling and the measurement approaches of these systems, or the characterization approaches. So just sort of a brief background on how we typically characterize these systems. We usually do some sort of radiative transfer analytic modeling. And there are a lot of different radiative transfer codes out there, but the two main things that they depend on when it comes to this regime of scattered light is some dust density distribution and some treatment for the scattering properties of the system. So this is just showing a bit of a cross-section view of a debris disk and some of the key parameters that are usually modeled for when it comes to characterizing some of these systems. And they mostly have to do with the size of the disk, the location of the disk, as well as the vertical properties of the disk. And that's just in terms of the density. So that's fine. But where things start to get really problematic is understanding the scattering properties of these disks. So some more background. The scattering phase function describes the distribution of light scattering around the circumference of your disk. So on the front side of your disk, let's say that your scattering angle is close to 0, and that increases as you go around to the back side. I don't think this is a super informative diagram to help you understand like what it takes to actually measure the scattering phase function or what it actually like really describes. So I made my own little PowerPoint explanation of what you can actually measure and what you can actually see with the scattering phase function, depending on the system that you have. So let's pretend that this is a completely face-on view of a debris disk. It has this nice ring shape. Photons are coming from the star, and they're deflected by the dust grains to reach the observer. So you're the observer, and you see this face-on disk. Now, when it comes to the what degree of scattering you see, all of the photons that reach you, the observer, have to deflect 90 degrees in order to see you. So that means that in your scattering phase function, and this is just a figure showing measurements of scattering phase function, in a face-on system, you're only ever able to probe 90 degrees of scattering angle. So this is all you see in a completely face-on system. Now, if you were in a completely edge-on system, so let's say you, the observer right here, in the sort of resolved image of the disk, you can see a lot more of these deflection angles or these points. So, directly in front, that's 0 degrees. Your photon goes here and has to deflect 45 degrees from incidence to get to you. And then similarly right here, has to deflect 90 degrees from incidence to get to you. And depending on the inclination of your system, you can also start to probe some of these backscattering angles as well. Hopefully that helps explain the scattering phase function a little better. Now, the scattering phase function is really important because being able to probe all of those different scattering angles can give you a sense of the sizes of your dust grains, as well as the composition, because different compositions and different grain sizes and different combinations of those will have different scattering efficiencies as a function of the scattering angle. Now, usually when we model these, we typically do a couple simplifying things to help us understand what could be generating this scattering profile. One of the most commonly used ways is the use of this Henry-Greenstein function, which is purely an analytic functional form, but it's not physically bounded. So there's no reason to expect that dust scattering properties follow this functional form. It's just a convenient and easy way to actually, fit for a scattered light profile. The other way you can do this is you can actually parameterize some of the properties of the dust grains themselves. So the composition, the shape of the grains, the size distribution of the grains. But as we've been seeing from all of these studies that we've been doing in high contrast imaging land, none of these formalisms actually work very well, and it's incredibly degenerate. And so this is a figure that I'm showing from Arriaga et al. 2020 that is trying to fit this measured scattering phase function of a debris disk and also trying to fit a measured polarization fraction phase profile simultaneously with some of these different dust grain models. And there is no combination of compositions or grain sizes between either of these grain shapes. So the mean scattering assumption or DHS, which is the distribution of hollow spheres assumption, there is no model that can fit both of these profiles simultaneously. And this is not new. This is known to be a huge problem that mean scattering and other very simple dust grain modeling approaches are not good. So what can we do about this? Okay, so this is another figure that I made of just showing you how much your choice in the scattering phase function actually matters. And so this is just an inclined disk model. Generic, this is sort of following, I'll explain what generic is in a couple of slides. This is a measured scattering phase function from a debris disk around HR 4796A. And then this is isotropic scattering. So yes, this is to just drive home the point that your scattering phase function choice and your brightness profile does matter when it comes to your modeling.
I guess, but you're calling the observational profile a scattering phase function.
Yes.
But is that the intensity or the scattering phase function?
Yeah, so in your images, so there's a difference between like your surface brightness and your scattering phase function. Your scattering phase function describes like, the scattering intensity in like a completely forward scattering direction. So it's not quite the same as like as a surface brightness profile where you might just like plop down some apertures along your disk and like measure how much intensity you're getting from there. It's related to that, but you need to do some manipulation and deprojection in order to actually get to like the forward scattering properties.
Right, so when you're calling this the scattering phase function, you're saying that's the derived scattering phase function, given a set of other assumptions about
Right.
Isotropy across the ring, temperatures, eccentricity, all of that.
Right, yes. And it is dependent on, most critically, it's dependent on inclination, but it's also dependent on what you decide on in terms of like the vertical structure profile, because that is related to inclination, as well as, even your radial size as well, depending on, again, your inclination.
So there's even more degeneracy as they go in.
Right. Right, yeah. Yes. Okay. I don't have a lot of time. So to get around this issue, what I did in my modeling was, rather than use these Henry-Greenstein phase functions or model these dust grain parameters, what if we took a bunch of measured scattering phase functions from solar system dust, and we fit a function to all of that, and use that for every single debris disk model that I make to try to characterize these systems. And the reason why we can do this is because we've actually been noticing these similar shapes in solar system scattering phase function measurements for a long time. I'm going to skip over this because this is just the part where I describe how I do my disk modeling, since I don't have a lot of time. But essentially, I'm really interested in morphology, and so that's why I'm fixing this scattering phase function treatment, and I'm doing this sort of MCMC approach to find my best fit morphological parameters for my disk models. That's a summary of this. Skip over this. I'll get straight to the results. So I looked at 8 different GeoPy image debris disks, and again, I used the same scattering treatment for all of them. And it turns out I got pretty good fits for most of what I saw. The few exceptions I'll point out are these ones outlined in blue, and I'll get to that in the next slide. But these are highly asymmetric debris disk systems. And so what I'm going to point out in my next slide is that the reason why I'm probably not getting good model fits for these systems is not because of the scattering treatment, but because of the morphological model that I'm using. And then the other system where the fit is not as good is this one down here, and I'll get to that one in a couple slides. But like I said, the morphological model itself is somewhat limited. It's a symmetric disk model, and so if you have a symmetric disk model, you're probably not going to get a good model fit to a highly asymmetric system. But what if I just split each of these systems in half and model each side separately, just to see if the scattering properties can still work here? And they do. So in this limiting assumption that I fit each half of the disk separately, the scattering profile still appears to work pretty well for these systems. Now, the other system in that green box, that one actually has a measured scattering phase function from Millie et al. 2017, and that's this dashed gray line right here. This is very different from what we see in the solar system dust scattering phase function measurements. So unsurprisingly, my generic scattering phase function treatment doesn't work as well here. But what if I took that measured scattering phase function and use that in my model of the system instead? And when I do that, I get a much cleaner result. So this is trying to illustrate that, okay, maybe if you're interested in understanding the morphological properties of these systems only, you can use these sort of rigid or fixed scattering phase function treatments to handle all of your scattering properties of your disk. And then I'll just get into some of the population analyses that I did here, because that was one of the main goals of what I was trying to do. And so here I'm plotting on the y-axis. You can think of this as like a vertical scale height. And on the x-axis, you can think of this as like a relative thickness in the radial direction of the rings. And you can map these to different sort of dynamical sculpting scenarios. So in these sort of like bigger scale height regime, and maybe a more narrower ring width, you could think of this as possibly coming from perturbations from planetary sculptors. In this sort of low scale height regime, like down in the bottom here, you could think of these as like, maybe there's some gas in these systems that might be inducing drag that is preventing some of these dust grains, from being vertically excited in that direction. The other thing that we can look at as well are eccentricities and sort of the narrowness of what rings. So what I'm showing here in this red line is from a paper by Grant Kennedy, 2020, where he looked at some ALMA observed debris disks and found this, found some systems where highly eccentric systems were pretty narrow in terms of their relative ring widths. But we're not quite seeing that in this scenario here. So like we still have this sort of positive correlation trend, but it's not as strong as the Kennedy results here. And I'm really running out of time, so I'm not really going to go over too much of what's next, but I talked about polarization and polarized intensity imaging. We have an even bigger sample of polarized intensity image debris disks, and this is something that I'd like to tackle next. In particular with this sort of fixed scattering treatment approach, but it's a little more complicated because polarization profiles are not quite similar between different dust compositions and dust grain sizes as what you see in unpolarized light. So this is showing a bunch of laboratory measurements of polarization fraction curves of different grain size distributions. But maybe that's a good thing, because if we can fit these types of profiles and we can try some of these different profiles out, maybe we can actually start to say some things about the dust grain sizes or the compositions in these systems. And I'm out of time, so I will just leave up my conclusion slide and take any questions.
So while switching, we will have one question, and after both talks are over, we'll have more time to interact. And Justin is here at Stewart, so you know that you can actually ask him more. So stay here longer if you have more questions to discuss. But just one question. Anybody? Okay, now let's, can you?
Yeah.
He has a question.
Stop share.
Yeah.
So, okay, hey, thanks for the talk. I have your empirical way of using scalable phase function. Does it perform on an overall? When looking at similar systems like tens or thousands maybe of systems, does it perform on the average of such a theory? Or is it just, because you pointed out, yeah, there's of course still some unique systems which you would have to independently, but this is like the same with me theory. It's like a one fits all.
Yeah, so there are a couple of reasons why I think some of this works so well. So first is the region of scattering, or sort of the range of scattering angles that we're sensitive to in most systems. So I have it on a backup slide, but I can't show it right now. But in laboratory measurements of dust grain scattering properties, what we see in unpolarized light is that for the most part, the general shape of the curve is pretty similar beyond like 30 degrees in the scattering angle. And so the most differences actually occur in the very forward scattering area. But in most systems, we're not going to see that, because if we look at these systems in high contrast imaging, for example, these would be like behind the coronagraph. Or, you know, a lot of the systems that we have are not at the right inclination to be able to actually probe those regimes. So that's one thing. The other thing too, yeah, I haven't had a chance to really test it on a lot of systems because some of this approach is pretty limited by having sufficient signals to noise in your data. And so there are a lot of, there's a bit of a long discussion that we could have later about, you know, high contrast imaging, post-processing, and being able to get good signal to noise for your images. But I think that's a bit too long of a discussion to have right now. But basically, you know, not enough high signal to noise data sets to be able to test this approach out on.
Okay, thank you, Justin. And we'll move on to the second one. So Jerry, would you like to introduce Ralph?
I'd love to, yes. It's a great pleasure to introduce a very old friend of mine who first overlapped as postdocs in Cambridge in 1981. And we have known each other all along for most of the time since then. And in addition to being a delightful person, Ralph has also made a huge reputation for himself as an expert on planet formation and disks and also magnetic fields and interactions of disks and magnetic fields. And he's moved on to galaxies recently, which I'm delighted to tell you about. He got his PhD from UBC just before he went to Cambridge as a postdoc. And then he worked at Berkeley with, I knew that after. Who was it? Chris McKee, thank you. And Colin Norman at Baltimore before he moved to McMaster, where he's been there for a very long time, establishing himself as one of the pillars of theoretical physics, astrophysics in Canada. And he also founded the Origins Institute, which I want to mention, at McMaster. He was director of that for a long period of time, but you've recently stood down from that. Is that right? And that has established itself as with a great reputation for a really broad scientific approach to the origins of the universe, all the way through to the origins of life. Anyway, I don't want to spend a long time talking about him. I'd like to give him a talk.
Jerry, thank you very much for that super kind introduction. It's a delight to come here to visit many friends and scientific leadership here in many fields. It's great to be here with you today. My topic today, origins of biomolecules, information molecules. Before I get started, let me just acknowledge my great set of collaborators and students that I've worked on, whose work will be mentioned in my talk today. First of all, Ben Pierce, who's PhD with me a few years ago, will join Purdue faculty in just this summer. And several students from collaborators at the Max Planck Institute, that will be with Thomas Henning's group, in particular Callum Mulder-Kearney and Klaus Paschek, and then support from these agencies in Canada. So before I get into the question of biomolecule formation in different kind of critical environments, let me just give you an overview of those environments and what's important for this problem. So first of all, I'm not a biologist, but this is something that every physicist should know about life, which is the way we see important subsystems in what we call life and our life can be shown nicely in this slide. We need water as a solvent. We need some sort of information polymer, which will be the highlight of my own talk, membranes to separate one medium from another. That will be lipids, a source of that. metabolism, some sort of source of energy, and a stable energy cycle that extracts that, catalysis, etc. So these have been viewed as separate components in the way that we've talked about this problem. Clearly, this needs to be integrated into a whole, but as a physicist, we're going to take this apart into pieces. And the motivation for that is this result that most biochemists, I believe, in the world subscribe to, which is that RNA genes copied by RNA catalysts form the idea of an RNA world. The discovery in 1986 that RNA acts as a catalyst. And so we can actually have a self-consistent system here without external enzymes, that RNA itself and forms of it can catalyze its own production. And this has driven a lot of research on the origin of the RNA world. First of all, questions is, how do you make the feedstock molecules, the very bases that you make RNA out of? Question one that I will address. And here, hydrogen cyanide plays the crucial role. So you might think it's very complicated, but actually hydrogen cyanide, take 5 hydrogen cyanides together, you can make adenine. So the chemistry question devolves in, where do you get hydrogen cyanide from? And we can track it from there, which is what I'll be doing in this talk. Second question, how do you build an RNA polymer out of these? And for polymerization, the first step in the chemical world is the random polymerization of chunks, little bits of perhaps a few N-mers joining together, et cetera, randomly heading towards some sort of template-directed process as an intermediate step. And life itself has perfected machinery, such as shown here, which is ribozymes as the kind of tools by which modern cells make and splice and cleave and do the functions in a ribosome towards making, catalyzing reactions, building proteins, et cetera, and replication in the modern genetic code.
Ralph, before you go on, just for the
Yes.
biologists and chemists in the room like me. So is HCN you said it is the basic feedstock, is that the only molecule you need, or there's HCN and other things go into?
So I'll show you a few other things.
Okay.
But the chemistry basically devolves on how you're making hydrogen cyanide.
Okay, that's everything else easy by comparison.
There's whole networks of calculations involved, but that's the essence of it, okay? So here, just in my review, RNA itself is held together. Just think of it as a skeleton. Skeleton is alternating sugar, with 5 carbons and phosphate groups forming this backbone. The linkages that you need to form between the 3 prime position on this ribose and the 5' carbon on this neighboring nucleotide joined together by this phosphate. Okay, so the question is, how do you form such bonds? And in the absence of enzymes, and we're talking prebiotic world, where does that bond energy come from to make this structure? Here are the bases, by the way. Notice adenine, there's 5 nitrogens and 5 carbons in those rings. Okay, so that's your 5 hydrogen cyanide molecules. The bond energy could come from thermal energy, say in wet dry cycles, in which during the drying phase, you remove water, which actually takes nucleotides. If you take water away, you'll get this join here. Life does this with enzymes, removes water and uses energy to put these things together. In this kind of situation, it might be thermal energy, or a lot of groups currently around the world use this sort of an activated so-called oligonucleotides, where you attach an energetic group here somewhere to provide the chemical energy to do the job. Nothing that's in current life, but so this Shostak's group, et cetera, use this kind of device.
What about the temperature, like cold and hot?
Temperatures here, I'll be showing you models in which we've worked all of this out, okay? Temperature should be between 40 and 80 degrees. So my talk will focus on these 2 environments: exogenous delivery, and this case, endogenous synthesis. That is, protoplanetary disks are extremely good in making hydrogen cyanide and the steps leading to the formation of planetesimals and the meteorites, which are rich in these biomolecules, which are delivered to the Earth by these mechanisms. Secondly, the endogenous synthesis. We have planetary processes known as the Miller and Urey synthesis as an example, in a hydrogen-rich atmosphere in which we might be having the conditions to make hydrogen cyanide and the chemistry that follows from that. So my talk, the bulk of my talk, will focus first on this and then head to this. Okay, we've worked on both sides of this problem. Just for timelines, this is a review article we wrote a few years ago with a few of our students, in which the formation of the Earth itself does involve in this delivery of materials from outside. So there's a period after the formation of the sun, the formation of the Earth at 4.544, the moon impact, maybe at this range in gigayears. We do have evidence from the mineral record of the existence of a hydrosphere that would have happened by about 4.3 billion years ago. There's evidence from lunar cratering that there was still perhaps a heavy bombardment of materials back to 3.9 billion years ago. So this has been the formation of the planetary environment plus this delivery process. So the source of molecules for life, there are two processes operating here, perhaps simultaneously, generating the important molecules. Evidence for life coming from the biological side, perhaps 3.7 from the relative ratio of 13 carbon to 12 carbon. Life does not like to push heavy things around. Life prefers light isotopes. So if you look at isotopic anomalies, material that has less amount of carbon-13 is probably biological in origin. And so biologists use that as a tracer. So we have evidence at 3.7, just after this bombardment. Stromatolites, here we get into evidence from biology, but there may be a window here with certain in which life probably occurred somewhere very near this very violent epoch. We will make the case that it should have appeared much earlier. So just a few words, since it's a mixed audience, I expect, on just habitable terrestrial worlds. This is astronomy now. But for what might be the exoplanetary analogues of the Earth is, of course, a topic of great interest. This is one of the best collections of data that I could find in literature, in which astronomers have identified, by using the RV method, identifying masses of planets, transit method to deduce their radii, put these together, and you can make models. Here are the data with very well controlled error bars here, some of the best in literature. Normally these are very, very broad, but these authors have called a collection in which you lay down models shown here in the dashed yellow, which is a silicate, magnesium silicate composition on which Venus and the Earth lie. And this line of rocky planets with very little atmosphere stretches to about 25 Earth masses here. And then there's a break. You see at about 5 or 6 Earth masses, suddenly we have a wide range of possible planetary types here. Some of these might involve steam atmospheres, some might involve heavy hydrogen helium envelopes. If it's a very heavy hydrogen helium envelope, we've seen that we calculate that the greenhouse effect, the opacities from such atmospheres is so large that these are unlikely to be moderately cool planets. So this is the realm that might cut off Earth-type life formation in this very narrow range of planetary masses. And if we then search for planets of this type, which of those planets are in a habitable zone around the star so that water remains liquid? Here we've got some great examples. Habitable zone around the solar system, of course. Here's the Kepler system with one planet in the habitable zone on its host star. And in this recent survey, if you instead of looking at the mass semi-major axis for orbits, you plot here instead of the solar flux, instead of the semi-major axis, this green bin here It indicates planets would have this insulation of our Earth, that is liquid water. And if you tie this off at about 5 Earth masses, there's a very few planetary candidates that count as terrestrial abodes for our own life. This institute here you must have lots of talks on TRAPPIST-1. I won't say very much about it, except looking for atmospheres has turned out to be a tough job on these planets. Whether they even exist or not is an open question. The theoretical work seems to be pretty skeptical that there are atmospheres, even on the planets in the habitable zone, because of the intense X-ray and far UV activity of the host and dwarf stars. So whether this will pan out or not, if not, then the most common star, which you might expect to be the abodes for the largest number of planets in the galaxy, may turn out to be uninhabitable. We'll see. This is a most striking and important result, I think. So to my 2 topics now, exogenous delivery. We've done a lot of work on, so here we have a disk population, and then these disks, what we want to hunt is for evidence for hydrogen cyanide and other molecules such as methane. The reason we want to do that is to start the chemical process by which incorporating hydrogen cyanide into planetesimal type bodies, wherein they will undergo chemical processing into the biomolecules. So this is a great result from the MINDS program, which indicates this is a first detection, as far as I'm aware, in the inner planet formation region of such a disk, where you have evidence for hydrogen cyanide, which you can see in the bottom trace in yellow. For online people, if I can get my arrow, that this is in this region right here, which tells you that hydrogen cyanide is present in these disks and should therefore be found on frozen grains and should therefore be incorporated into materials out of which we build planetesimals. The route to building planetesimals, so let's imagine our grains here are covered with these important ices, methane, hydrogen cyanide, etc. What is the fate of such systems? We've got experts in this audience, Andrew and others, who work on this stuff and understand the evolution of dust in these systems. But to make a long story short, an aggregation process, the physics of the disk tells me that we can build dusty, fluffy dust grains up to some scale at which they start to break apart because the impact velocities may be too large. The turbulence dictates then how large these dust particles can become. After that, we have pebbles that are drifting through the disk to build larger structures. You can't make structures that are stable or very long lasting in this range. These are very lost to drag processes in the disk. So all current thinking, whether it's been completely verified or not, is that we skip over this range and go directly to the building of planetesimals, 100-kilometer sized bodies. So these are presumably trapped the chemicals from the disks that we're interested in. So let's look at these. Meteorites are chunks of these planetesimals, and they come in 2 varieties, quite stony, and then these carbonaceous chondrites, rich in organic compounds. As you see, Murchison, huge reservoir of these. 3 of the 5 nuclear bases are present here. Interesting question, why not all 5? Source of fatty acids, nuclear bases, amino acids in plenty. With my student Alyssa Cobb a number of years ago, we just searched through the literature to compile a list of the abundances of these amino acids in various kinds of these carbonaceous chondrites. The one characterizing Murchison would be in this class here. These are grouped by glycine down to tyrosine. Glycine is the simplest. It's always the most abundant. And with in a paper I did years ago with Paul Higgs, we showed that there's a thermodynamic reasons for these relative abundances of these molecules as they become thermodynamically harder and harder to build. So the chemical process within planetesimals, you can have radiogenic heating of these bodies up to temperatures of a few hundred degrees centigrade. This is perfect for synthesizing the reactions by Strecker reactions for amino acids, in which you take hydrogen cyanide, water, and an aldehyde, say formaldehyde, cook them, and you get glycine. That's the simplest case. Okay, so these very, very simple molecules put together in processes that we use in industry to make amino acids are very apparent in these planetesimals. So think of planetesimals as failed attempts to make life. Did they make RNA? Well, that would have broken down long ago, but it's an interesting thought. So the first results for nuclear bases are that there's a series of different kinds of simple chemical reactions which we identified are in the literature, about 60 or 70 of them that we called out, that can make these nuclear bases. This Fischer-Tropsch, again, takes simple feedstock in the presence of catalyst, and you make all 5 of these. Non-catalytic reactions, you just take hydrogen cyanide, heat and cool, with some water, you get all 5. Catalytic is the so-called formalism reaction. I won't talk about that. A few catalysts, you're successful. Let me show you. Let me just focus on a few of these reactions, because this is how we built our chemical networks. Just take adenine. Here's a list of reactions in the presence of, these are Fischer-Tropsch. Here's non-catalytic. This is the one I highlighted, 5-hydrogen cyanide in water gives you adenine. Uracil, there's the role of hydrogen cyanide as an example. Keep going down the list. That's where your nitrogen is coming from primarily. Put all these together and calculate this at the temperature regimes, densities that you set in the planetesimals. In our first paper with Ben, we tried this, assuming that the abundances of hydrogen cyanide in these molecules were that cometary abundances, the only data we had, would be a good place to start point for your initial abundances of these simple molecules. Later work with our MPIA collaborators showed that, in fact, if we're talking about planetesimals in the near solar system, you should take the chemical abundances, say, in the asteroid belt, use those, do a detailed calculation of the interior of the planetesimals, and find the dominant reaction. We can calculate interior of these bodies, where the various, this is for adenine, where in this body it's being formed, because there's a temperature gradient in the body. Fairly detailed calculations come to the following conclusions, that if you use comet abundances, you're going to overproduce things that you don't see. So if you use data, say, from the abundance of materials expect from the inner solar system, where you've got asteroids, maybe even inside the ice line, but near to it, then you get pretty good matches for adenine and guanine. We discovered that if you try to make cytosine, it rapidly converts to uracil. It disappears very quickly. So how you continue to make cytosine and thymine is an interesting question, how you do that. What degrades them is interactions with water. Get rid of that amine group, the NH2 group, which destroys the capacity. Yes, let me just, we did a very detailed calculation now on the fate of such materials delivered to the planet. This was in a paper in PNAS in 2017. This is some of the most detailed calculations you'll find in the literature on the fate of delivery of materials to a planet into warm little ponds. So what can happen to you here, here's our basic molecule. And in a dry wet cycle, because we're setting ourselves up for the conditions now, what conditions do we need to get polymers to form? Wet dry cycles. In the rainy part of a cycle, or the wet part, you have a pond here. Analyze pond distribution sizes to do these calculations. We had bombardments, rates derived from lunar cratering records. Put all of this together, we understood how meteorites break up as they come into the atmosphere, typical size of pieces here, precipitation rates taken from modern Earth measurements, etc., and infall of dust particles indicated by Chiba and Sagan thought that this was the predominant form of organic deliveries through of this interstellar dust. And in the dry part of the cycle, the pond is gone. You're now exposed to UV, which breaks down these molecules, deaminate it, takes off this group. In the wet part of the cycle, water interacts with this to knock off this group. So the destruction mechanisms are UV radiation, it's hydrolysis, it's loss of material through the porous bottom of the pond by seepage. You put all this together and the resulting calculations are that we have these fact that islands are rising out of the sea, continents gradually are moving up. And so in our models, we actually compute the available target area, that is the number of ponds that are on the surface available to be hit by these fragments. Bombardment rates are, of course, many billions of times greater than we have today. This is the formation of the Earth period. So putting all that together, we find that the meteorites could deliver levels to about parts per million of concentrated bases in these ponds. The gradual seepage of this material out of the meteorite is important for being able to feed the pond for maybe up to 100 years or so. The dust particles, they mix, the hydrolysis gets them very quickly and destroys the biomolecules. And so this hot early Earth environment favors the rapid nucleotide synthesis in these models. And but the fact is that this is a highly transient formation mechanism. So I will, do I have a few minutes? What do I have?
Yeah, 2 minutes, yeah.
A Mitch? Oh, I should end at 1?
Yeah,
Oh, okay.
but a couple of minutes.
Then let me just point to this. So this process was discovered by Miller, etc., Stanley Miller and Urey, in which you're making hydrogen cyanide in a reactive, highly reducing atmosphere, hydrogen rich. Hydrogen, if you pass electric discharge, that's your excitation energy, and that you break down things like the methane molecules in your gas. The ammonia breaks that up into radicals that react to produce hydrogen cyanide. Once you have that, you produce everything else. That's the way this experiment works. So you need a highly reducing atmosphere to do this job. So this is a view of how hydrogen cyanide works, chemistry in an atmosphere. Just give me a minute to go through this. So there's evidence that the material on the surface of the Earth was oxidized already by 4.3 billion years ago. That's not a good environment for making these molecules. You have impacts moving in. Large impacts, iron reacts with water to produce ferrous oxide plus hydrogen. There's your hydrogen. You can maintain that for a while. So in the hydrogen cyanide part, methane, acting on by UV and lightning, will break up into radicals, CH2, CH3, your nitrogen similar into individual nitrogens. That methane fragments radicals together with the nitrogen makes hydrogen cyanide. That rains out into pools. Here, the aqueous reactions, think of those flasks in the Miller-Urey experiment. This is where you're actually making the biomolecules then. The methane that's important is a key component of these calculations. The methane arises from the subsurface geochemistry here, these plumes that are rising. Hydrothermal plumes produce a lot of methane here. And the result is here. So this is a plot with time and abundance of these various materials. This orders of magnitude change here, but you can see the meteorites produce a burst of amino acids in your nuclear base in the pond, and they decay fairly quickly. Depending on environments, you won't get this longer than a century or so, whereas this steady infall over 200 million years before the atmosphere becomes oxidized, over 200 million years, you can maintain a parts per million level of this material in your ponds. This oscillation has to do with your pond wet-dry cycles, by the way, to drive the formation of these different species. And here we've got uracil, guanine, et cetera. So we can calculate the resulting concentration of these materials in our ponds fed by this source. So I will just quickly go to this slide. I'm going to end my talk saying we've built a reactor, a laboratory with a planet simulator, which we can alter the environment in here with wet dry cycles, different radiation fields, all cycled in this regime in order to study how we might make polymers in prebiotic conditions. We're hoping for a run this summer, in which we got our first real results. But I'm going to leave it there. Thank you very much.
Question, George.
So it's always seemed to me the problem with building RNA is it's a complicated molecule. And there's a time scale issue, and you referred to that in one of your slides about how stable everything has to be along the way. But do you think this is still an open issue, that in all the environments things are, that they're still not obvious how the timescales work out so things are stable and you can actually get there, or is that a solved problem?
I wouldn't say it's solved, but there are various routes. So you're absolutely right. What plagues RNA and plagues life, actually, in our own cells, is that hydrolysis is always destroying stuff. So cells are physically expending energy to repair that damage. And cells have a way of protecting molecules by putting things in membranes. A membrane walks out water. So probably an important piece to this is two things, is probably the presence of lipids that spontaneously make membranes. And having this synthesis occurring inside sort of a simple lipid bag for my cell might be one way of mitigating this problem. And in fact, if you talk to David Deemer, who's been one of the advanced people in this field, they imagine that this evolution, it's a co-evolution between a vesicle, a bag, and the RNA that's in it. And I think that might be a general way of solving this, but there's no general solution yet. And it is a key problem.
Caitlin?
So you mentioned, that the composition of planetesimals, whether you should use like asteroid belt or something else, but I'm wondering about the Earth itself. Is the idea that differentiation just destroys any HCN that you're born with, and that's why you have to wait for it to be delivered, or?
Well, the Earth itself is continuously making it. So when we did all this work for the meteorites, people said, sure, Ralph, but we've got the whole Earth here, and surely we can make molecules in this way. So it was kind of a challenge to us to actually calculate what the yields might be in realistic environments. That was the challenge. And it was a bit of a surprise to us that the yield that you get from the planetary process, it's like the peak yield that you get from meteorites, except that it just kind of continues along as a steady source. We still think that this is too low a yield. And so the important aspect here, maybe I could point to one slide, which I skipped, which is this calculation right here, which is the relative abundance of methane, which is the important thing, compared to hydrogen cyanide. And no matter what environment, this is a reducing environment, this is an oxidizing environment. Oxidizing environment is way low on the yield. Just follow these two purple curves here. And you see that the hydrogen cyanide, the lower purple, always follows this curve by a few orders of magnitude down, no matter what you do. So the driver here may really be, what is your source of methane? And methane is this geochemical process I indicated before. The more methane you get, that's by a separate reaction, a water rock reaction that produces hydrogen, and that reacts with CO2 down there, making the methane. So the question is, what is that underlying submantle process that's churning this up? And what is the methane yield for that? So our current work that we're doing, we're working with a Caltech postdoc on revised models for continental drift and tectonics to understand how much ferrous oxide can you dig out, iron can you dig out to react with water to make hydrogen, et cetera, drive. So that may be the critical rate determining step. We know already we could get orders of magnitude more methane if we change those models somewhat. So that's what we're on to at the moment.
Yeah, so.
Yeah, one last question, yeah.
Yeah, so yeah, you talked about the fact that there's all this evidence for complex organic chemistry in asteroids that are delivered as meteorites to bases. I'm wondering what, right, the asteroid belt is obviously inside where astronomers say the CO ice line is, right? And so presumably you had to have, right, carbon-rich ices from the outer solar system, right, delivered during the formation of the asteroid belt. So I'm wondering what what these models of what's required for this chemistry, right, how that can be inverted to tell you the initial composition of asteroids.
Yeah, well, we're that's the planet formation part of myself. That you want to do this link between the astrochemistry of disks, one point, how the dust migrates in such disks, which is work that you do or the work that's done here by several of you, and where does this? What is the composition of the material that ends up in this terrestrial planet zone? That is this combination of astrochemistry beyond the ice line and how much of it you deliver inside the ice line where the terrestrial planets actually form. So for that, you need, several kinds of things, which we're working on separately, but that's an open question.
Well, this chemistry is obviously very complicated, but you seem to be saying that water is kind of the enemy. But then, right, in comets, right, water ice is far more abundant than any other kind of ice. So how are you doing this on a planetesimal with all this water in the way if it's a problem?
Right. Well, the planetesimals were successful, right? Clearly, the structure synthesis seems to get the right yields. So this is in making the nuclear bases. Okay, so there is an issue. The planetesimals do dry out. The temperatures fall with time. After 10 million years, you're done. So it may be that last gasp as you're starting to freeze material that you're left with the abundances that you see. The timelines in Paschek's calculation take into account the normal water abundances. So yeah, it's you need the aqueous reaction to make the thing. It's an aqueous reaction after all. But once you've got the nucleotide, it can also work against you. So it's relative timescales, and the chemical synthesis timescale is very fast. The hydrolysis is slower, but it's interesting. And it's the balance between those rates that will determine it.
All right. Anybody who has a burning question from online? So thank you so much for both speakers today. And let's thank speakers again.